All right. Is that the right distance for the mic? Can everyone hear me okay? All right. I just want to start by thanking Mātai, the whole team, Sam, for this great event. It's been a real privilege to be asked to come and present here. And I met Sam, I think, about 5 years ago now when she came and gave a talk at the ABI. And was really impressed with the work and her interest in MRI. And so it's been a real pleasure to be able to watch Mātai be sort of conceptualized and then launched. And I've been down here collecting a lot of data since it was launched, and so it's been really great to be a part of it all. So I want to start my talk just with a little bit of historical context. So the study of biomechanics is trying to understand the way that biological systems work, the way that tissues work, the mechanics of living things. Biomechanics has, for a very long time, been rooted in really understanding the anatomy. And for actual millennia, going back as far as 500 BCE, anatomists and biomechanists have been interested in basically dissecting the muscles of animals or cadavers to look at them and try to understand how movement was produced. This is something that people have been really interested in, and understanding how the musculoskeletal system produces movement, because it's actually the sole tissue in our bodies that allows us to move. And so, such profound actions that make us human and make us animals come down to the musculoskeletal system. And so for a long time, people have been performing very, very careful dissections, across cultures and across history to understand the nature of the tissues and how they work. And it was about a little over, I guess about 125 years ago now that medical imaging it enabled this kind of paradigm shift in our ability to understand anatomy and therefore understand biomechanics. And so we go from this painting, which shows an anatomist, a medical professor doing a dissection of a cadaver, to that's basically the first X-ray, as well as an early MRI photo, showing that we can actually study the anatomy of living people and understand what their tissues look like in vivo. And so it sort of moves us from, on the bottom row, this cadaver data, and many cadavers are sick when their bodies are donated or they're elderly, to being able to generate these in vivo digital models of the anatomy from medical imaging. On the right side, another area that's really interesting to me is tendons. And so medical imaging allows us to go from these very, very simplified models of tendons to a fully 3D realistic finite element model that's informed by the in vivo anatomy, informed by imaging. And so just pivoting a little bit to talk about the tissue that I'll be talking about today. So I'm really interested in connective tissues. So I started out my career studying skeletal muscle, and skeletal muscle is a force-generating tissue, which is really interesting. But it turns out that in order for anything to come of those forces, they have to be transmitted to the bones and the joints. And the tissues that they're transmitted through are these connective tissues. So tendons, these big, thick, cord-like structures that carry the force from your muscles into your bones, and aponeuroses, which are a long word, but it's basically these sort of flat tendons that connect the muscles to the external tendon. And so they're these sort of sheets that wrap around your muscles, and the muscle fibers will insert into those sheets. And so understanding the nature of these tendons, how they store and transmit energy, is actually a real key part of understanding force transmission in these structures. So this is just showing you some of the tendon and connective tissue anatomy in the hand. The hand is full of connective tissues. If you know anyone who's a rock climber, they'll tell you all about their connective tissue injuries in their hands. It's a very common problem and needs to be a lot of time taken to restore tendon health after an injury. This is a model that we created some years ago, and this is showing the complex tendon anatomy in the triceps suri. So that's the muscle and tendon structure in the back of your calf. It's hugely responsible for movement, and it's associated with the Achilles tendon, the largest and strongest tendon in the body. And then moving on from tendons and aponeurosis, the fascia is another really, really interesting tissue. Fascia is this connective tissue sheath that encases muscles, and it likely has this role in force transmission. One of the reasons why this is really interesting to me is that for a long time in musculoskeletal modeling, the fascia was just ignored. So we would develop models of muscles and how they transmit forces into tendon, and this other tissue, the fascia, just was kind of left off of models. If you go back into the literature as far back as the 50s, 60s, and 70s, or even before that, people were talking about how abundant fascia is in the body. So it interdigitates all of your muscles, it encases and surrounds your muscles. It's this really, really common tissue, but we were generally leaving it off of models when we were trying to understand motion. So just showing you a few images of that. So you can see the fascia encasing muscle on the left. If you do dissection, you see these sheaths with fibers that run in multiple directions through the fascia. And as you dissect a body, the fascia is just ubiquitous. Surgeons and people who've trained in surgery will tell you that it's the fascia is everywhere in the body when you start performing a dissection or a surgery. Just a little bit of foreshadowing. That's an MR image that we've collected that shows the fascia in good detail and in high contrast. So just a little bit more about the importance of this tissue. So I really like this image because it shows the fascial structure, which is it's huge and it runs through the entire body, and that the fascia image sits between the muscle and the bone. So you've got the skeleton on the right, the muscular tissue on the left, and the fascia right in the middle. Now, a lot of people think this is wrong because the fascia encases the muscles, but what I like about this arrangement is that I think the fascia is a key player in how forces are transmitted from the muscles to the bones, and so I like that it's in the middle there. Some people have even said for a conceptual understanding of fascia, rather than thinking about we have many, many, many muscles all throughout our muscular system, you can think of it as we have many, many fascial planes that divide up our body, and you have one muscle that resides between all of those many fascial planes. But it's quite interconnected, and we think stores a lot of elastic energy and probably a key feature in how the body moves. Moving on to talking a little bit about MRI and how conventional MRI is acquired. This diagram that I've made is showing what's called K-space. That's the domain in which MR data is collected. It's basically frequency space. We're collecting resonating frequencies in the scanner. And conventionally, how we do that is we move from the center of K-space, so the center of those frequencies, we move out in X and Y, and then we collect along each of those lines, along each of those readouts. And so a lot of the MR hardware was sort of designed for this acquisition, and it works quite well. The problem is when we're trying to get connective tissues, so tendons, they just turn out completely dark when we do one of these conventional scans. And so I've shown the Achilles tendon. It's just that dark structure there. You can't really see any detail in it. So yeah, here's another one. So the Achilles tendon is this dark patch that's on the back of the ankle. This is an ankle image. And here's another one. So the Achilles tendon in that left image would be this dark patch that's running through, or that small little oval on the bottom of that image. The problem with trying to understand a tissue when it's dark in MR is that you're getting no signal from it. And so you could say, well, it's the tissue we're getting no signal from, but there could be many adjacent tissues that you're also not getting signal from. And so it's hard to distinguish the tissue that you're interested in. And so in order to solve our problem, we've moved to what's called non-cartesian imaging. And in this case, I'm showing you a radial acquisition. So rather than starting in the center of K-space and moving out and selecting along one readout, we're actually just sampling right from the middle of K-space and just sampling right away from the middle, moving out. There's some challenges with non-Cartesian imaging. One of them is reconstruction, because we have to reconstruct the X and the Y frequencies simultaneously. But nevertheless, if you can overcome those challenges, it gives you the ability to sequence to image the tissue really, really rapidly. And so that leads us to what we call ultra-short echo time, or UTE images. So if we can sample really, really quickly, we can actually image the tendon before that signal decays away. And so just showing you that, so this is an ultra-short TE image, and so you can see the tendon there, rather than being black, is gray. And so comparing that to the conventional short TE image, the tendon's black, we don't get any data from it. But what's really clever is that rather than just taking this ultra-short TE image, we can actually do image subtraction. So we subtract the second image from the first, and when we do that, we get this really, really high contrast image of the tendon, where the tendon's showing up bright white in this picture. It gives us extremely high contrast and signal to model these tissues. So just showing you a little bit more of that. Again, here's a UTE image of, so this is a sagittal view of the Achilles tendon, and that's a short TE image. And you might be saying, well, I can see the tendon, great in the second image where it's dark. There's a contrast there, so what's the problem? You can also see it in that image on the left. But one of the challenges is what you probably didn't see from either of those two images is that the tendon is surrounded by dark tissues in this subtracted image, and that's actually water and bursa that surrounds the tendon. I've seen a lot of models generated from of the Achilles tendon from these conventional images, and generally the tendon shape is incorrect in a lot of these models. Some of these models have been shared hundreds or thousands of times with people who are doing biomechanical modeling, and so I think it's really important that we nail the imaging so that we can generate really, really good models of that tissue. So here's another subtracted image just to give you a sense of what we're interested in. This one and this final one actually shows you the fascia, that really important tissue that for a long time I've thought was important that we can generate medical images of. Because if we can get images, then we can develop models without having to go through the dissection step, which disrupts the tissue, and it allows us to have this pipeline, including fascia, where we go from imaging all the way to generating models. So using this approach, again, we can visualize aponeuroses and tendons really well. These are just some more images that we've collected over the years, over the last few years. And so on this very last image, I'm highlighting some aponeuroses. I'm highlighting the Achilles tendon in different locations. But on this very last bottom right image, you can actually see this bright white sheath that surrounds the muscles. And that bright white sheath is the fascia. And so to our knowledge, this is the first time that anyone's been imaging the fascia tissue in vivo. And I have a PhD student, Rondika Pereira, who will be publishing the first, to our knowledge, database of fascial thickness and extent throughout the body. So we're able to get some anatomy that hasn't been generated in vivo and document what we're finding. So just to show you a little bit of the modeling that we're doing, so this is an anatomical image at the top, and our MR image here on the bottom left, and so just showing that we're able to capture the anatomy in vivo in high detail, and by identifying the fascia, so that thin sheath that surrounds the full anatomy, we can develop this computational model. This is a geometrical model, and we can then start performing the mechanics, so computationally understand how the mechanics of the fascia is interplaying with the muscles. So physics-based simulations of the dynamics between those tissues. So we're currently working with Mātai to develop what are called MASDIR protocols. So this is just another way of taking advantage of UTE and image arithmetic. And when we do that, we can see all kinds of really interesting tissues that are bright, so high signal and high contrast. And so it's really opening up for us the possibility of including these connective tissues in our models. We're also going to try to integrate this with diffusion tensor imaging. So what I'm showing you there on the right is from imaging, but those are actual muscle fibers that are in a person's lower limb, and we can basically develop these very, very high throughput, high resolution models of muscle and connective tissue anatomy, how those interact, and how the mechanics work. So I just want to acknowledge funders and collaborators, the Mātai team who I've been working with for some time, and who really are just a delight and a privilege to work with. Thanks.